So before I start with the actual presentation, I have to declare one potential conflict of interest, because I'm also an external advisor and animal welfare officer for a medical device company. But let's get to the important bit, namely the DECIDE project. DECIDE stands for Decision Enabling Confirmation of Innovative Discoveries and Exploratory Evidence, and we're doing this with quite some people here at the Quest Center. And before I go into detail about the call, just a little bit of background information. I don't think that I have to tell you a lot about the reproducibility crisis or lack of translation, because I guess we wouldn't be here if we were not all aware of this. However, yeah, just a quick reminder of where to place these confirmatory studies. Typically, evidence generated in preclinical efficacy studies is weak, so we have often low numbers of animals, selective reporting, publication bias, and also consequently inflated effect sizes that might threaten the translation. And the idea is that this preclinical evidence might be strengthened with confirmatory studies that are basically proposed as a tool to weed out false positives, gain deeper understanding about mechanisms, and refine the experimental design based on the exploration by increasing validity and reliability of results. And a little bit more now about the call. So this was a pioneering call from the BMBF 2019 to 2018 called Confirmatory Preclinical Studies and Systematic Reviews. It consisted of overall 3 modules. So one module about these preclinical studies, obviously, another module focusing on systematic reviews, and third module would be the DECIDE project, the accompanying project for the whole call. And the aim of this funding measure is to strengthen preclinical research and its transfer of results, and the confirmatory studies are intended to validate the findings of exploratory studies. And overall, 12 studies were funded, and importantly, all multi-center studies, so no single lab studies. This is, of course, not the only initiative when it comes to reproducibility or replication studies. There's also, I think, one of the more famous studies, the Reproducibility Project Cancer Biology, but also the Brazilian Reproducibility Initiative. However, what I think is important and what I would like to highlight is that in the case of the BMBF call, researchers are incentivized to validate and confirm their own findings. And I think this comes already sometimes with a different motivation and maybe also even like different challenges. So how does this look like in practice now? So as I mentioned, it was 12 confirmatory preclinical projects. All of those projects were across Germany, with very few exceptions, where centers in the multi-center study had to be in different countries. It's varying biomedical fields. It's different species. So we have mice, rats, but also large animals like sheep or even non-human primate studies. It's all different medicinal products. So from vaccine studies to drug development, there's a little bit of everything. All of those studies had to be pre-registered, and importantly, each had statistical advice, and each needed to decide for a total sample size for the multi-center study. And we, as the DECIDE project, had with each of the projects one-on-one consultations to get an idea what are potential needs or also issues. And what we realized very fast, and this graph I took from our 2 PhD students on the project, is that there's a lack of guidance. If we look into the exploratory versus confirmatory effect sizes, you can already see that almost every project used a different approach, and this is only like one part of it. So what we are trying and still working on with the researchers together is to develop some sort of guidelines. So this is why I'm also presenting some lessons learned from the DECIDE project. So one thing that we did to develop some of those guidelines is to host and design workshops on prerequisites and design of these preclinical confirmatory multi-center studies. We also included Tim Arrington from the Cancer Reproducibility Project as a keynote speaker, and we had some guiding questions. With the overall guiding question, what is actually feasible for practitioners? Because paper might be patient, but we also have to account for limitations in the actual laboratory work. So one question was, what evidence is actually needed before I can start a confirmatory study? So what are really like prerequisites, and how can I plan a confirmatory study? We also looked a little bit into analysis and what is actually confirmation success, but this is beyond the scope of this particular meeting. So I skipped that for now. And what we came up with when we look into a preclinical research trajectory and have an exploratory study, what we typically aim for to have some sort of robustness check. So we see whether studies are of high validity and reliability. If yes, then everything is fine. You might be able to go and plan a multi-center confirmatory study. If no, you might want to go back and have, for example, a small within lab replication to actually strengthen your preclinical evidence. So how can that be strengthened then? So you have a hypothesis generating study, and if the uncertainty is still high, optimization of this evidence can be done via replication studies to basically increase the sample size, improve internal validity, but also introduce systematic heterogenization and or also flanking experiments. So basically the idea is also in that line that the exploratory study should inform the confirmatory study. So you need a high degree of internal validity, meaning blinding, randomization, so on and so forth. And you can use your exploratory study to identify, for example, confounding variables, to have a block design with your randomization, for example. Then it's very important to really a priori define inclusion and exclusion criteria. You have to have a well-defined primary outcome, and you have to have some idea about what kind of statistics you want to apply. And this is, of course, all things that can be written down in the pre-registration. And just a small image on that, why it's so important also to have inclusion-exclusion criteria and also some sort of outlier management. If you look into that graph, I think it becomes very clear that you should always look at all data to define outliers and not separate by experimental groups, for example. So, and ideally always report full data sets with all excluded animals, ideally with the reason why you excluded them. Another thing that is very important, or that we figured is very important when we plan confirmatory studies, is that we need to increase the reliability and so increase the sample size for the confirmation. So you should either consider effect size shrinkage for a power calculation, or you should basically aim for something that we call smallest effect size of interest. And this smallest effect size of interest can be informed by clinician. It can be a biological relevant effect size. So there can be many ways how to come up with this. You just need to transparently report this. The other thing is, of course, with respect to external validity, everyone was very clear that there should be no way around using both sexes unless you are working maybe with prostate cancer or something that is very sex specific. That's a different story, but other than this, there should be no excuse why you shouldn't use both sexes in those preclinical studies, or also include potential comorbidities or else. The other thing how you could increase your external validity and generalizability is a refinement of the model that you use. So sometimes it might be feasible to use a complexer indication. Then with respect to translational validity, it makes sense to include biomarkers or also diagnostics from standard clinical care, and also in general have a closer look into the clinic, whether there is already a competitor or not, and if yes, you should compete with that treatment that is already in the clinic, and not only consider a naive control group with your treatment. Because you can also imagine, if you look into effect sizes, for example, that an effect from treatment comparison to naive control comparison will most likely be larger than a treatment 1 versus treatment 2 effect. And then an important challenge came especially with the fact that there is all multi-center studies. So there are several labs involved, and they are not like right around the corner, but in different federal states all over Germany, or even outside of Germany, for those some examples I gave before. So what is considered very important and also lesson learned, you need to early identify difference across centers and also come up with training. So especially if you're doing behavioral tests, all of those things, you need to have a certain level of training to account for inter-rater reliability. One thing, and this is also for us still a little bit work in progress, is the question about standardization versus heterogenization, and how to really balance this. So if any of you has some ideas there, please drop us a line. So we are constantly still working on that and trying to figure this out. And with respect to protocols, that means, so not only if you're doing a single lab study, but especially if you're doing multi-center studies, you need to harmonize protocols, procedures. You have to look for naturally occurring differences when it comes to housing. You might want to have systematic heterogenization to a certain level, and you might also need to look for quality control, quality assurance across centers. If you transport something back and forth, how do you get your drugs? All of those things. With respect to experimental design in all centers, bias should be really minimized. It might make sense to have a visualization of the experimental design, and you want to be very clear again about inclusion, exclusion criteria and your outcome measures. Can also be very helpful if you have a multi-center study. One lab might have another expertise than the other one, so it might be feasible to have flanking experiments or triangulation from that lab and have additional evidence that can then be still exploratory, but be part of the confirmatory design. And of course, when it comes to anything that has to do with analysis or sample size calculation, I think the main recommendation that we had is really to consult a statistician. And what we also realized, not every lab, not every university really has easy access to this kind of expertise. So when we overall look into what we have learned, I think one main thing, and this is also important for the meeting about the Good Research Practice Toolkit, is that especially with confirmatory studies and everything that focuses on translation, we need some sort of minimum validity and reliability criteria before we should even take that next step. So we need to have a certain quality of the study to go a step further. That said, there's always the opportunity to optimize the evidence via other application studies. It's very important to have standardized protocols in place. It's important to look into the model system. Is it relevant? Is there any sort of limitation that should be acknowledged? Sample size calculation, smallest effect size of interest, or shrinkage of the effect size from exploratory studies should be considered. And you might want to introduce sources of variation. This goes back again to systematic heterogenization and what I mentioned just the slides before, all of these multi-center considerations. And maybe also one lesson learned, and this is one of the DECIDE outcomes also at the Berlin Institute of Health from the Quest Center, that we also started now a whole unit, namely responsible preclinics, where we try to tackle exactly that issue, that most of the time one research lab doesn't really have the infrastructure, so that they don't have necessarily a statistician or someone who could advise with regulatory affairs and all of those things. And for example, here at the BIH, we already have something that I would call a translational ecosystem, and responsible preclinics will now connect those different stakeholders. We provide also project-specific targeted counseling, so same as we did and learned from DECIDE, because this was also mirrored back to us that this was the most helpful, that we really had these one-on-one sessions and really took our time to discuss and went and dived deep into literature and had joint workshops and all of those things. And what we also try is to develop and evaluate a metric, not only to assess, but also to identify action items with those projects. So what can and cannot be improved. And then also, of course, always assess and question ourselves whether we are asking even the right questions and doing everything in an appropriate way. And with this, I would like to thank you and, of course, highlight that this is a team effort and not just me presenting this. So if you have any questions, you can, of course, contact me directly. So you can see my email address here. But you can also contact the whole team via our DECIDE email address, decide@charité.de. And if you would like to know more about a guide that we wrote as a recommendation for a second BMBF call on those confirmatory preclinical studies, you can use this link. And if you want to know more about responsible preclinics, we also provide the link here. And with this, thank you again, and I'm looking forward to a successful meeting.